So for this, I'd like to introduce the chair, moderator, and speaker in this session, Dr. Rommie Amaro. Rommie holds the Distinguished Professorship in Theoretical and Computational Chemistry at the University of California, San Diego. She's the recipient of multiple really prestigious awards, including NIH New Innovator Award, Presidential Early Career Awards for scientists and engineers, and others. And her scientific interests lie at the intersection of computer-aided drug discovery and biophysical simulation. Rommie, the floor is yours. We're excited to hear this session.
Thank you, Serene. Let me go ahead and share my slides. Okay, hopefully you're starting to see those. Okay, so can you see everything? Look okay?
Yes.
Okay,
that's great.
It's great.
Thank you.
Yeah, no problem. Hello, everyone. I'm happy to be here. I think it's been so far a really great 2 days, and we're sort of bringing up the caboose now. And where we wanted to really focus is in bridging the gaps in translation. And we have our session today. I'm going to give a little bit of an intro. Everybody will get 20 minutes, including myself. And then Pat Walters from Relay Therapeutics is going to give a 20-minute perspective on dangers of poor data. Russ Altman from Stanford University will give a 20-minute perspective on large language models and their evaluation of regulatory submissions. Then we have Joel Karpiak from GSK, who's going to be talking about team environment for better translation. And then we are going to go into, we'll go directly into a panel discussion. And with all of us here, including Marty Head from Amgen, who was one of the opening speakers yesterday. So it should be good. What we're going to do is actually let folks give 20 minutes and that 20-minute sort of talk. And then instead of having Q&A, we're going to save all the Q&A directly for that last 40-minute session. And I also want to say to invite folks and remind audience members to please submit questions for that, because I think what we want to do is have a good discussion about topics that are of interest to folks who are attending, and maybe get insights that aren't necessarily part of like a typical presentation. Okay, and then I have to confess that when I was listening to everything, I was getting so excited for the science because I can't help it. And instead of just sort of, because we're going to have 40 minutes at the end where we can yammer all about and discuss about different challenges. So I wanted to actually give a little bit of a view about what we are working on in this space. And I think it's hard to follow David Baker. It's not the first time I've been in this position, darn it. And now it's his Nobel Prize. Congratulations, David, again. But he did mention at the end, and it's come up, what about dynamics? And dynamics is something that myself and my group's been super interested in. We've heard a little bit about it. Especially what we're interested to do is use data-centric, physics-based molecular dynamics simulations as a so-called computational microscope. And so this means that we can integrate all different types of experimental data sets coming from different modalities. They could be different structural data sets taken at different resolutions, like X-ray, cryo-EM, cryo-ET. There could be lipidomics, genomics, et cetera, glycomics. We can combine all of these different data sets taken in different groups from different repositories, and then build these these systems, these atomic level representations of biological systems that really, are sort of allows for these sort of cohesive understandings of drug targets. And then all we're simply doing is approximating that system down to its many atoms. We have that potential function here that basically describes each of the interactions with all the other atoms in the system. And I think John might have talked well, actually, I'm not sure if he talked a little bit about this functional form, but there's a lot people are doing with machine learning to try to sort of update this functional form, which folks have been using from the 70s. But in any case, then we generate often tons of data, could be terabytes of data of just sort of structural dynamical data sets through the integration of Newton's equation of motion over time with these atomic coordinates. And so the analysis is another place where, there's a lot of just sort of course coding that has to take place, but now where machine learning is starting to come into that. And then, of course, we visualize this and sort of interpret the experiments so that we can advance our understanding of biology, including the development of new small molecule drugs, for example, that combine cryptic pockets that aren't present in X-ray crystallographic or cryo-EM structures, because we know, all of these targets are dynamic, and often they do open up new pockets for small molecule discovery. What I wanted to tell you about today are our efforts more on protein design. And it's true, as David mentioned, there's been a huge focus on small molecule design. He went to binders, small protein binders, and I'm going to actually take it a little bit further and talk about how simulation can allow us to see the unseen for targets for immunogen design. And so, I don't want to spend too much time on just the method, but, this is really now sort of a multiscale computational microscope, actually, that lets folks probe drug targets and their environments from the quantum all the way to the macro scale realm. What I want to talk about today are some of our efforts just in the nanoscale. And so the example that I just want to tell you about quickly is with the SARS-CoV-2 virus. We all know about this. It's a lipid enveloped virus, and many of us have had. It has structural proteins like the E protein. There's a few of those tiled inside the membrane. Then you have the M protein. There's hundreds of those on the inside of the viral membrane that provide structure. And this is an example of one of the structural models we built for the whole virus. We and many folks are interested in these viral glycoproteins, including the spike protein, which I'm showing here, because it sits on the outside of the virus and it plays a key role in infection. But it also is a key element of immunity, because that's where a lot of our adaptive immunity responses are directed. So we built a comprehensive model that included sort of an integrative model, I should say, that included both cryo-EM resolved data sets for parts of this, as well as used predictions for other bits that were not able to be experimentally resolved. That includes adding the sort of extensive glycans that are present in these systems, which are also highly mobile. There's also the transmembrane part, so really the entire end-to-end sequence is represented at the atomic level. And then we can, like I was saying before, animate the dynamics. And so this is how we get at these sort of proteins. This is really, fortunately or unfortunately, computing and simulation is really the only way to get at these high resolution dynamical data sets. I mean, there is time resolved cryo-EM now, but you will only resolve some states, and that's still sort of a work in progress. Simulations allow us to view the dynamics of these systems with relatively high fidelity. And so we have been interested. So again, going back to the spike protein, most of what we have designed is actually targeting the entire spike protein, which actually includes that S1 domain. So the spike protein has the S1 domain, which just sort of popped off there. And then that actually peels off during the process of infection, and then is left with the S2 domain, which is shown here in the middle. So this is really sort of the inside core of the spike protein. And so we joined a team of researchers, Jason McClelland, Karthik Chandran, et cetera, and tried to target S2, this inside core, potentially for pan-coronavirus vaccine development, because we all know that very unfortunately, S1 mutates like crazy. And so the most conserved parts of this beast are actually on the inside core of the protein. So S2 is highly conserved across the beta coronavirus family. So it's been thought that it would be a potentially good target. It also has immunogens inside there, some at the top, some immunogens I'm showing here in red, also in yellow, and you have one in the stalk that are there. So it's also immunogenic. One of the big challenges has been the instability, though, of this. So, you express it, if it's in an mRNA construct, it just basically sort of falls apart. So it has the prefusion S2, where you have these stabilized epitopes, is actually unstable. And this is the case even for these really sort of highly designed and optimized stable hexapro mutants of S2. They also just sort of flop apart. So really further stabilization is required in order to use this as a pan coronavirus antigen. And so this is where we wanted to bring in molecular simulation. And not only did we just do, it's not just, in this case, what we wanted to do was actually use sort of a more advanced, or what we call an enhanced sampling technique called weighted ensemble molecular dynamics, which is a way of overcoming one of the central limitations of the method of MD. So one of the issues or challenges with these methods is that they are physics-based and they're slow. So computing those equations is rather slow and is computationally intensive. Ideally, we would have enough data that we could build fast surrogate models with ML, but we're not there yet. And so we and many others in the field are in the process of generating high quality data sets that can be utilized by machine learning and AI approaches, but we're not there quite yet. So in any case, we turn to weighted ensemble MD, which is an enhanced sampling. It's called a path sampling approach that basically helps to get us out of these basins. I lost my mouse, but that's okay. It helps get us out of these sort of wells that we're usually stuck in with dynamics by spawning many trajectories and then following the ones that are sort of successfully progressing across a predefined reaction coordinate, in this case, which was sort of the opening of the apex. So here is the S2 subunit, and what I'm about to show, so you can see it's a trimer, because the spike protein is a trimer. Generally, many of these class 1 glycoperfusion proteins are trimers, and what we did was we used weighted ensemble to basically explore sort of the opening pathway. So our thought was, and our strategy was, if we could learn something about the mechanism of opening of this potential immunogen, could we then use information about the mechanism against it in order to basically stop that progression from happening? And so here's just another view of this molecule sort of progressing along what we define as our reaction coordinate. So we start up here in the upper left quadrant, our closed conformation, and we're progressing towards the opening state. And, when you run these simulations, you actually get many ensembles of pathways. And so a lot of work has to be done to try to tease out which is the dominant pathway. But in any case, we can view that. And so by looking at that opening pathway, what we saw was when we looked closely at the mechanism of opening, we could see that it sort of unzipped in an asymmetrical manner. And that when we did a closer analysis, what we found was that if you look at the central helices sort of stratified like a ladder, as I'm showing here, with the helices in purple and the residues in yellow and orange, what you could see is that there were particular positions, in particular that top one, V991, and then the fourth one down, T998, that actually did not make really strong contacts. And in fact, when you look at the opening pathways, you could see that those were sort of quickly kind of falling apart. So what we did then was actually we learned the mechanism, and then we actually just tried putting in tryptophan mutations into those locations in both that top rung as well as the fourth rung. And when we did that, what we found was a highly stabilized network of interactions between the different, between not only like each of the sort of central helices, but also there's sort of network of interactions between the residues that are sort of underneath it, as well as going down to the fourth level and above, and then even an outside network of stabilized residues further. And you can see it's like it became, so we actually made this one suggestion, or these two suggestions, and then simulated those. And when we saw that it actually stopped the opening, or it didn't stop the opening, I shouldn't say, it dramatically slowed the opening relative to the construct we had been working with. We then handed it to Jason McClellan and his team, and they were able to express the double mutant. What they saw was that it was actually, I'm sorry, I lost my pointer. they saw that actually, if you look at differential scanning temperature plots, that actually these two mutations created a highly temperature stabilized construct. In fact, the most stable that we know of now. And the other cool thing was that because of the flexibility of this construct initially, they were never able to get high resolution structures of just the S2 because it was just too dynamic. However, with these two mutations, they were in fact able to get the high resolution structure, which was really cool. And it validated all the findings that we had from our simulations and predictions about what the effects of these mutations would be in terms of sort of the structural integrity in the pre-fusion state. We then passed this to Kartik Chandran and his team, and they immunized mice, and then did the SARA experiments, and they found that it was just as good against these recombinant viruses against the original Wuhan strain, but actually had improved performance versus Omicron. And actually, with these binding affinity interactions, that are shown on the right-hand side, we actually saw that these epitopes were still being recognized and bound to. And so there's a lot of folks to acknowledge there. I still have a few minutes. I just want to say, so I feel like this is sort of a little bit of a different tack than what we've heard, where instead of running, I'll say one of the advantages of this technique is that instead of running like tons of wet lab experiments, it's sort of, I think, a little bit of a different view where you can take a deeper dive into understanding a target and its mechanism of action, its dynamics, and then, in fact, be highly strategic about which mutations you're actually making. So I think there was a sort of a large efficiency. They created the double mutant and then each of the single mutants, and then we had these results, which were, sort of where we got to the milestone that we were sort of looking for. That said, of course, and as David also mentioned, there's a lot of challenges in this field and for the field in general, for drug and immunogen design generally. And we're going to have a chance to talk a lot about these in this last session, as we've been discussing, I think, a little bit along the way. But certainly, with the PDB, which enabled AlphaFold and, all of the great sort of fruits of that labor, which took many years in the discipline of, a whole field and a real sort of commitment to sharing data and the format of it and so forth. We now see how that needs to reverberate across many fields, including, protein dynamics, including ligand, small molecule structure design. And so in my closing thing, I see, Pat, you're ready to go. I have maybe 2 minutes, so I just want to just touch on a couple of challenges here, just to sort of like see some of the discussion that we'll have in a minute, and then I'll flip it to you. But as one of the things that we are particularly interested in, again, which I guess maybe kind of a little bit unique, but it is in sort of enlarging sort of the scale and the complexity of systems that we're able to simulate. And there's, with this, we have such amazing data sets for so many different targets and coming at them from so many different directions now, really being able to, I see a lot of challenges related to that, and also with data. And I think data is kind of like, in some ways, a funny thing, because it's like, well, we have a lot of data, but then it's like, oh, maybe it's not the right type of data. And then, especially in our field, what we see is that there's a lot of, and in biology in general, you still have a lot of cottage laboratories generating data, but it's not like necessarily unified or being shared in a way that can be, used. And I know I'm not the only person that's used in AI and ML, going forward. Of course, there's hardware limitations. Even though there have been great advances in GPU computing, there are specific limitations for some of these dynamical algorithms that I think will need to be addressed. And then there's a lot of social aspects, and I'm sure we get to touch on this in the last little panel discussion. And, there's different social aspects depending on academia and industry. But I think the field as a whole, drug discovery as a whole, that whole ecosystem will do better by sort of trying to address some of these. And then, of course, validation and interpretation, which Pat is probably going to talk a lot about. Maybe we get to hear one of Pat's rants, one of my personal favorite things to do.